Hi, my name is Martin Bushell and I work at the MRC Toxicology Unit in Leicester. We work on how microRNAs control gene expression. MicroRNAs are small non-coding RNA molecules. There's about 2,000 of them in our genomes, and they control, at some stage or another, about 50% of all protein encoding messenger RNAs. MicroRNAs go wrong in a large number of human diseases, particularly cancer, but they don't just go wrong, they're actually part of the driving force that leads cancers to develop. So microRNA-based therapeutics are becoming a real possibility, and the first clinical trials are in humans are going forward at the moment. But how do these molecules work to control gene expression? And that's what we've been really interested in examining over the last few years. And we've got our recent papers coming out in Science, and this shows that microRNAs control gene expression at a very early stage in the process. They control it at the initiation of protein synthesis. This control at the initiation of protein synthesis is not just happening, it's required to happen for microRNAs to function, and that's what we show. We also then go on to show precisely the factor that is required for this control, eIF4A2. And what this is doing is it's being recruited into the microRNA repressed messages via interactions probably with the CCR4 NOT complex. This recruitment of this factor is inhibiting the progression of the ribosome from finding the start codon on these mRNAs. And this is of fundamental importance because what we find is that mRNAs have to have secondary structure in their 5' UTRs for microRNA-based regulation to take place. When we look at genome-wide analysis, we find that by bioinformatics, we find that microRNA target sites are more frequent in messenger RNAs that have more structured 5' untranslated regions. So it's all starting to tie together. So what we believe is taking place is that this eIF4A2 is being recruited via the microRNA repression machinery to mRNAs, and this is stopping the progress of the 40S ribosomal subunit from finding the AUG start codon. And this process is required for subsequent mRNA destabilization that takes place following microRNA repression. Anyway, thank you very much for listening.